Welcome to today's Explainer. I'm Dr. Chem, and I am absolutely pumped to take you on a trip right into the biochemical engine room of yeast fermentation. Picture what you're seeing right now in this animated laboratory setup. Tiny yeast cells furiously gobbling up sugar molecules, releasing energy, and blowing out bubbles of carbon dioxide. Today, we're going to unpack exactly how that happens. We'll explore how this beautifully simple biochemical process, where yeast converts sugar into ethanol and carbon dioxide under completely anaerobic conditions, has fundamentally shaped human history and continues to drive massive global industries today. Here's the roadmap for our explainer today. We're going to start with humanity's oldest domesticated organism, then break down the actual chemistry of fermentation. After that, we'll test some fermentation variables, dive into a fascinating glucose versus sucrose debate, look at oxygen's role with the Pasteur and Crabtree effects, and wrap up with bread, beer, and biofuels. Let's get right into it.
Part 1. Humanity's oldest domesticated organism. So take a look at this. It might just look like a bunch of dormant little spheres, right? But this is Saccharomyces cerevisiae, up close under a scanning electron microscope. This microscopic fungus is, no joke, humanity's oldest domesticated companion. We've been working with these guys for millennia, way before we even had the tools to know they existed. To put that timeline into perspective, yeast fermentation has been happening right alongside human civilization for a very long time. We're talking about brewing in Sumeria and Babylonia around 6000 BCE. By 1300 BCE, the ancient Egyptians were baking bread, basically treating rising dough like it was pure magic. It actually wasn't until 1680 that Antony van Leeuwenhoek finally spotted them through a microscope. And the real game changer? 1857, when Louis Pasteur literally proved these living cells were the engine behind the fermentation process. Moving to Part 2, the chemistry of alcoholic fermentation. So, stepping out of the history books, how does this microscopic machinery actually work? Well, in 1897, a chemist named Edward Buchner did something wild. He ground up yeast cells, pressed out the liquid, and found that this cell-free juice could still ferment sugar. He had isolated an enzyme complex he called zymase. This was absolutely crucial because it proved fermentation isn't just some mysterious life force. It's a biochemical reaction. It's just a clever way for cells to extract energy when there's no oxygen available. Let's break down that machinery step by step. When yeast meets a complex sugar, like sucrose for instance, it first uses an enzyme called invertase to cleave it into simpler pieces. Then, a process called glycolysis snaps that glucose down into pyruvate, which gives the yeast a quick, vital hit of ATP energy. Next, that pyruvate gets converted into acetaldehyde, which releases that famous carbon dioxide. And for the grand finale, the acetaldehyde is reduced to ethanol. And here is the beautiful math of anaerobic respiration. We start with one single molecule of glucose, C6H12O6. The yeast works its biochemical magic, and out pops exactly 2 molecules of ethanol, 2 molecules of carbon dioxide gas, and, the whole reason the yeast is doing this in the first place, 2 units of cellular energy, or ATP.
Part 3. Testing fermentation variables. Okay, enough theory. Let's take these equations and head to the lab bench. How do we actually measure this? We start by activating dormant dry yeast in warm water. And you've got to remember, variables like temperature are huge for the fermentation rate. We then prep 5% solutions of our fuel, glucose, sucrose, and a water control in conical flasks. We mix our yeast suspension in, stopper it up, and run a delivery tube into an inverted graduated cylinder. Over 60 minutes, we capture the evolving carbon dioxide bubbles. By measuring that froth, we're literally tracking the speed of the reaction in real time. Part 4. The glucose versus sucrose debate. Now, going into the experiment, scientific textbooks will tell you yeast strongly prefers simple monomers, like glucose, over a complex disaccharide like sucrose. But we got a massive plot twist. Completely contrary to expectations, the sucrose treatment pumped out way more CO2 than the glucose. Over our 60-minute run, the sucrose fermentation spiked to over 35,000 parts per million. Unbelievable, right? But, in science, we don't just trust our eyes, we run the numbers. To make sure this wasn't just a total fluke, we ran a single-factor ANOVA test. The result? A p-value of 0.04. Since that's under the standard 0.05 threshold, it completely confirms there is a statistically significant difference in how our yeast process these different sugars.
So what does this all mean? Well, real-world experimental variance often completely challenges established theories. The literature says yeast loves glucose best, but our specific lab run showed a massive preference for sucrose. It really suggests that, hey, maybe we need a larger sample size, or we need to look closer at the specific strain of yeast we're using to definitively map population-level behaviors. Science is all about constantly testing your assumptions.
Part 5. Oxygen's role, Pasteur and Crabtree. Now, aside from the type of sugar, oxygen changes the whole game, leading to two completely opposite but fascinating biochemical phenomena. First up, the Pasteur effect. If you give yeast plenty of oxygen, it actually stops fermenting and switches to efficient aerobic respiration for maximum energy. Makes sense. But then there's the Crabtree effect. If you dump a massive amount of glucose on yeast, it basically panics. Even if it's swimming in oxygen, it stubbornly churns out ethanol. Why? Evolutionary biologists think it's a brilliant defense mechanism. The yeast ferments the sugar to poison competing bacteria with alcohol, basically hoarding the food source for itself. How cool is that?
Part 6. Bread, beer, and biofuels.
Let's zoom out a bit. The exact same microscopic chemistry we just watched in the flask powers a global industrial footprint. In the baking industry, we're harnessing that carbon dioxide to make our bread rise. In the beverage world, it's all about capturing the ethanol from grapes or malted grains. And perhaps most critically for our future, the biofuel sector ferments high-yield crops like corn and cassava to produce ethanol that gets blended right into gasoline. To really put the sheer scale of this single-celled organism into perspective, the global alcoholic beverage industry alone was valued at a mind-boggling $223 billion back in 2015. That entire economic juggernaut is driven by the exact same tiny biochemical reactions we just mapped out today. I want to leave you with this final provocative thought. If humanity's oldest domesticated organism has already built a multi-billion-dollar food and beverage industry just by breaking down sugar, what is the actual ceiling here? As we get better at genetic mapping and yeast biotechnology, could finding or engineering super-efficient yeast strains be the ultimate key to replacing petroleum fuels forever? The answers are quite literally waiting for us right there under the microscope. Thanks so much for joining me for this explainer. Keep asking questions, stay curious, and I'll catch you next time.